In our first video, we talked about the 3 steps of cellular respiration, including glycolysis, the Krebs cycle, and the electron transport chain, or the ETC. In our first video, we also went over the process of glycolysis and the essential things that glycolysis and its function do and give us. The first, of course, is 2 ATP, or the energy currency of the cell. We also got 2 NADH, and we also got 2 pyruvates. Now, going on to the Krebs cycle, you can see that the utilization of pyruvate is extremely important into furthering the process of cellular respiration. And you can see in the picture right below here that it's quite important in creating another substance that we'll get into. This process is called pyruvate oxidation. After glycolysis and the production of pyruvates, which of course happens in the cytosol, pyruvate actually goes inside of the mitochondria. Within the mitochondria, which is done through transport proteins, not only does it release a carbon dioxide, CO2, and an NADH, but at the same time is changed by coenzyme A, and pyruvate converts itself into acetyl-CoA. Again, this process is called pyruvate oxidation. Now, it might seem kind of useless at first that we're creating a small conversion of pyruvate to acetyl-CoA, and for many people, they simply gloss over pyruvate oxidation. However, the conversion of pyruvate into acetyl-CoA is extremely important, especially when it comes to starting the Krebs cycle, which is something that we'll get into now. Pictured here, you can see the entirety of the Krebs cycle, and at first glance, it looks quite appalling. There's so many structures, there's so many long names, but we really don't have to get into the specifics of the citric acid cycle, also known as the Krebs cycle. Instead, let's try and see what it's trying to do in the first place. Now, of course, at the very top, when I told you not to ignore pyruvate oxidation, this is the main reason why. We have acetyl-CoA right here, which is the direct output of pyruvate oxidation in the first place. Now, when you enter in acetyl-CoA, it acts as the starting point of the citric acid cycle, which ultimately goes through a bunch of processes, whether it be removing hydrogen ions or removing an oxygen, anything of the sort, to create and go back to acetyl-CoA once again. Now, we can see here through the processes, citrate, isocitrate, alpha-ketoglutarate, succinyl-CoA, we can see that actually here, between succinyl-CoA and succinate, we actually create ATP. And with that, the cycle continues on in a regeneration process to bring back oxaloacetate and use acetyl-CoA to create citrate once again. Now, as you can see, there are a couple main outputs of the citric acid cycle, including carbon dioxide, NADH, ATP of course, FADH. And that is all done twice. Because remember, for each turn we produce, these outputs right here, NADH, CO2, ATP. And remember that for each step and each time glycolysis is done, 2 pyruvates are actually formed, which means that we turn the cycle twice, which means that you get 2 times all of the things shown here. We can see 3 NADHs, multiply that by 2, and we get 6 NADHs from 1 cycle of glycolysis. We get 2 carbon dioxides in 1 turn, and 2 times 2 is equal to 4 carbon dioxides from the citric acid cycle being turned twice, and we also get 2 times 1 of FADH, which is just 2 FADH2s. Now, although it might look extremely complicated, the citric acid cycle can be boiled down into a repeating cycle which produces NADH, which is an electron carrier that we'll get into in the electron transport chain, 4 carbon dioxides, and 2 FADH2s. Ultimately, that's simply all it is, a cycle that creates processes. Of course, if you wanted to take your time to, it's quite simple to learn the actual substances within the citric acid cycle if you made them into a mnemonic, but for now, that's simply all you need to know. Taking something as big as the citric acid cycle, converting it into a couple of numbers, moving on to the next step of our energy creation process.